Welcome dear students to my channel Biology with Dr. Ishrat.
Today we are going to discuss fungi like Protista or Protoctista. As you know that these are the slime mold, they lack chlorophyll, they are surrounded by the plasma membrane only. Vegetative cell or vegetative phase lack cell wall, however the spores have the cellulose cell wall. At one stage of the life cycle, they have amoeboid structure, or it is also known as vegetative body plasmodial structure. The slime mold live usually among decaying vegetation. They commonly occurs on lawn and moist fields. They exhibit wide range of coloration. They are saprophytes and have phototropic mode of nutrition. Parasitic form are not known. Both asexual and sexual mode of reproductions are present. They produce spores within sporangia. A spore possesses cell wall of cellulose. The slime mold resemble both protozoa and the fungi. They are like protozoa in their amoeboid plasmodial stage and similar to true fungi in abundant spore formation. So fungi like protistas, they are protists that absorb their food from dead organic material, and they are grouped into two groups: slime mold and water molds. Here you can see there are different type of slime molds. It is chocolate tube slime mold, it is yellow in color, it is pink or purplish slime mold. Here you can see that the slime mold is on the leaves, and here is another color of the slime mold. It is white in shade. These are the different shades or colors of slime mold. So what is slime mold? Slime mold, many of them live as a single celled organism. Slime mold live in cool, shady and moist spaces in the wood or in fresh water. They eat bacteria, yeast and small bits of decaying plant and animal matters. They eat surrounding particles and then digest them. Slime mold usually measure about 1 to 2 centimeter, but a few slime molds are as big as several meters as well. They often have bright colors such as vibrant yellow, brown and white colors. So here are the types of slime mold. Slime molds having two phases, cellular phase and acellular phase. We will differentiate what type of acellular and cellular phase are there and how can you recognize that this one is a cellular phase and the second one is acellular phase. The first one is acellular phase or slime mold. It is also known as plasmodial slime mold. The plasmodium structure consisting of a distinct multinucleated mass and involve 10,000 dividing nuclei without any individual cell membrane. This colorful protoplasmic mass creep along in amoeboid fashion. You must have seen the locomotion or movement of amoeba. So it is also creep just like amoeba and over moist and cool and shady places such as within the cracks of decomposing wood, underneath or partially decaying bark of the trees, trunks and stump or other organic matter in high humidity condition, and in leaf debris on the forest floor which degrade and feeding by endocytosis. The other stage of its life cycle starts when plasmodium mass is starved. It means it do not have any food material, so it is in starving condition or in dried condition. At this time, plasmodium typically differentiate and develop fruiting bodies containing stalk and cellulose cell wall. Fruiting bodies are resistant to the environmental condition because they are the spore bodies and they contain spores, so they survive till the favorable condition resumes. The spores and wind dispersal occurs under favorable conditions. Spores germinate and haploid amoeba-like flagellated cells are released. These cells fuse forming zygote, then these diploid cells feed and nuclei divide to form multinucleate plasmodium. Here is the life cycle of acellular slime mold. You can see that this is the feeding slime mold and in maturation in dry habitat it turn into sclerotia. The sclerosias are also resistant to the environmental condition. When that condition become favorable, these mature plasmodium will germinate and produce young sporangia. And young sporangia, when mature, then there will be meiosis, and due to result of meiosis, haplide spores will be formed. These haplide spores will germinate, and then we have the amoeboid cells or flagellated cells will be formed. These two different haploid cells will fuse together by means of plasmogamy and syngamy, then zygote is formed. This is the life cycle of acellular slime mold, the slime mold which is a plasmodium like. Now we will discuss another type which is cellular slime mold. This group of slime mold has complex life cycle involving true multicellularity called pseudoplasmodium. Pseudoplasmodium which is different from the acellular slime mold as true plasmodium. It consists of aggregated mass of individual amoebas like structures that are able to migrate as a large motile multicellular slugs. Actually, when the star cell release molecular signals, then adenosine monophosphate and a specific glycoprotein, the other cells sense these molecules because they are starved, so they will sense those adenosine monophosphate molecule and aggregate around that signal producing cells. Ultimately, some of the amoeba cells sacrifice themselves and differentiate into the dead vacuolated stack that produce fruiting bodies containing countless and so many numbers of unicellular spores. Under favorable conditions, these spores release and germinate, and then the vegetative amoeba that feed on bacterial cells and yeast are released. After running out of food and starvation, the amoeba aggregate to form pseudoplasmodium again. As a life cycle of cellular slime mold, you will see that both type of life cycle, cellular and acellular as well. Firstly, this is the sludge or moving plasmodium. It will develop fruiting bodies. These fruiting bodies will mature and then they release the spores. And these spores are emerging amoebas because they will germinate to form amoebas. These free amoebas will again fuse together and they form zygote. Then here is the asexual reproduction. You can see that these free amoeba, they begin to aggregate together. They will join together. These are large number of amoebas like structures. They will aggregate and they will form this amoeboid form, amoeboid mass, and this is the asexual reproduction. Here you can see that sludge or moving mass, a mass of amoeba will develop fruiting bodies. These fruiting bodies, spores will form emerging amoebas. These amoebas will aggregate to form a amoeboid mass. And in sexual reproduction, you can see that these two type of haploid emerging amoebas will fuse together. They are free amoeba. Then they will fuse and they will form zygote, and after formation of zygote, they will form the new amoeboid stage. This is the life cycle of cellular slime molds. So here is the economic importance of slime mold. As you have studied that these are the basically harmless and are beneficial because they are also known as wonderful organism. Still, we cannot say that they have any type of loss to any human beings or any type of plant or any other living organism. Slime molds are not toxic, not poisonous or venomous. So they help to build up the sandy soil by returning nutrients to the cell because they engulf those nutrients and they feed upon the rotten leaves of the plant. So they again return those nutrients to the soil. Spores of slime molds move easily because they have come in contact with mulch. However, they are also have blown in from anywhere. Slime molds cause the decay and decomposition of organic material in the soil. They creep over the ornamental plants and make them look ugly sometimes. Attractive colors are of artistic values. The plasmodia of slime molds are an excellent material for the study and of structure and physiology of protoplasm. So here's the economic importance. Now we will discuss another type of Protoctista, fungi like Protoctista, which is water mold. Water molds. Water molds mostly live in water or moist soil. They can be parasite of plant and animal and getting their nutrients from these organisms and also from decaying of these organisms. They cause common problem to the farmers and they cause variety of plant disease. One of the most famous of these disease was a fungus attacked to the Irish potatoes, and that was Phytopthora infestan. This time, potatoes were the main source of food for many of the Irish people, so they suffer with the potato famine. So the failure of the potatoes crop mean that many people in Ireland died after starvation and migrated to the other countries. So what is Phytopthora infestan? Phytopthora infestan is an oomycete, or you can say that this is water mold and fungi like microorganism that cause serious disease in potatoes and tomatoes and other members of family Solanaceae, and they cause late blight of potatoes. Late blight was a major culprit in 1845 or 1840s in Irish and 1846 in Highland potato famines. The organism can infect some of members of the Solanaceae as well. As I have told you that this pathogen is favored by moist and cool environment because it needs 12 to 18 degrees centigrade optimal temperature and fully saturated water contents or environment which will be the fully saturated for spore production, for zoospore production. So lesions growth rate are typically optimum at slightly warmer temperature, which is 20 to 24 degrees centigrade. So here you can see these are the leaves of potato, or you can say Solanum tuberosum. These leaves firstly infected, then the mycelia goes inside the soil, and then your potato crop will be rotten or spoiled like this. These are the two pictures of potatoes. This type of symptoms occurs when the Phytopthora infestan affect the potato crop. So the life cycle of Phytopthora infestan is characterized by alternating phase of hyphal growth, sporangia, sporulation, and zoospore release, and then reestablishment of hyphal growth. There is also sexual reproduction or sexual cycle, which occurs when two opposite mating hyphae like Oogonium and Antheridium meet. They will form the sexual spores called oospores. The different types of spores will play a major role in dissemination and survival of Phytopthora infestan. Sporangia are spread by means of wind or water, and they can cause the different they affect the different plants, host plant. So the zoospores released from sporangia are biflagellated and they are chemotactic, so they move from one place to another place because of the attraction of that chemicals. So here we will study the life cycle of Phytopthora infestan. You can see that it is infected foliage of potato crop. Then this infected foliage, then the hyphae will reaches to the stem tubers. As you know that the potato is stem tuber. Then the infected tubers or infected leaves having the sporangium, and then that sporangium will form zoospores. These zoospores will enter into the leaves by means of stomata or by means of any opening in the cells. This is the asexual life cycle. While in sexual life cycles, you can see that the hyphae or the infected tuber having two types of different mating strains. Oogonium is a female strain and Anthridium is a male strain. Then both Oogonium and Anthridium will fuse together. They form oospores, and then oospores will form oosporangium. This is the sexual phase of Phytophthora infestan, and this is the asexual phase of Phytophthora infestan, and it causes great loss to the potato crop. So here are symptoms of Phytophthora infestan. Due to appearance of watery light green or yellow blisters on the leaf foliage, have you seen those the spoiled leaf or infected leaves? Then the lower leaves develop brown and black lesions. In certain areas of the plant, leaf blistering may occur within several days. It means generally 1 to 4 days leaf rotting occurs by the development of tan colored blisters and in dry weathers, and in blue-gray mycelium in moist weather. Later, leaves start falling and rotting in tuber also occur. Rotting in tuber are of two types: dry rot, which cause bluish or black and reddish brown fungal growth and develop outside and inside the potato tubers. Phytophthora infestan cause 5 to 15 millimeter deep rotting in tuber. You have seen already. And the second one is wet rot. The potato tuber develop the growth of white colonies outside the and secrete water. Phytophthora infestan cause 24 to 45 deep rotting in tubers, or we can say that it affects the whole potato. So how you can control the disease or Phytophthora infestan? By selecting the resistant varieties of the seed. The plant debris must be clean and maintain field sanitation. To control the lead blight infection, one should apply or one should use a sprinkler irrigation technique. It means the very small quantity of water you will use, then that will allow foliage to dry, and due to regular interval there will be the lowest chance of that disease, because you know that the Phytophthora infestan is water mold, so it require water as well as a cool temperature. So if we will not give the optimal or good conditions for that growth, there will be the lowest chances of sporulation of Phytophthora infestan. Harvesting potato must be done in the dry season. As I have told you that the dry season is very much effective to control the Phytophthora infestan disease. Excise the upper portion. If you see that there is lesions or there is the chances on the leaves, then we should excise. We should cut those leaves, and then before harvesting, then we will have the healthy crops. There should be balanced use of nitrogen fixation fertilizer, and then seed must be treated with 5% redomil, and dust of 1 kg of seed we will use that 5% of redomil. It will also decrease the chances of Phytophthora infestan. Seed should be stored in the 3.5 to 4.4 degrees Celsius. That infection should not be there. So the sources of inoculation like weeds and infected tubers must be eliminated. When we eliminate those sources, we will have the healthy crops. One can also use foliar fungicide. There are a lot of fungicides which will rusticate or which resist, or which will you will see the good growth of your potato crops, and Phytophthora infestan will be not affected that crop. So we will use the foliar fungicide as well. Thank you so much.
For more videos, like, share and subscribe my channel.